Welcome again to Let's Talk Forensics with Pastor Dan. We're going to do the study guide today. Test is coming up on serology, so make sure that we're ready to go. First question we have there is, what is the means of determining the relationship of a suspect to crime based on blood or how blood is found? Second question is, what is the first means of identifying blood analysis in 1901? Third question, what is blood identified? What in blood identifies the type? And then for the fourth question, identify the four types of blood. 5 is true or false. Antibodies of the same type of blood will result in clotting of the blood sample. Number 6, antigen what gives us the Rh factor? 7, list the proteins that can be found in the human body. Number 5, given the following percentages, and I have them listed there, identify the percentage of population that is B negative MN. Number 9, identifying clues based on patterns of blood found is called what? There are 3 lines there. The 10th question, it wasn't until 1955 that blood splatter was first used at a what? And true or false, the first case involves blood splatter at trial was used by the prosecution. 12th question, identify the 3 parts of blood. So I list those down there. Blood is found because it is blank and blank, the 2 aspects of blood. List the 5 types of blood splatter found. That's the first page. And the second page, here we go. List the 3 ways that blood can determine the point of origin. Identify the factors of blood drop size can tell us. Study of drugs that poisons found in the body is called what? True or false? Marijuana is the most abused drug in the United States. Number 19, explain briefly how a breathalyzer works. 22 blanks there is one of the first tests done on blood to measure pH. Number 21, list the 2 main items tested for drugs or poisons. Blank blank is the main instrument used to test for drugs or poison in blood. True or false, carbon monoxide poisoning result in discoloration of skin tissue. 24, name the main question in determining of blood. 25, list the 2 main testing of human blood. 26, blank reacts with iron in the human blood. Number 27, the main problem. This test is what? There's one thing we mentioned. 28, the measurement of pH. What measures the pH of the blood, which is usually human blood around 7.5. And the last question then lists the 4 other types of tests for human blood. So go through, answer those questions. Again, there's the first page, and there's the second page. First page, pause it, screenshot it, whatever you want to do. Second page, all right, pause the video, answer the questions. Alrighty, here we ready? Here we go. All right, so the first one is blood analysis. Second is blood typing. Antigens A, B, O, and AB. It's false. It's called glutination. D, MM, MN, NN. The percentage is 0.864%. Blood splatter analysis, trial. And I'm not sure I mentioned it, but it was used as part of the defense. Red blood cells, white blood cells, and plasma. Cohesive and absorbent. A pooling, gravitational, directional, cast off, and spray. Following the blood trail, lines of convergence and void. Height of a fall, type of weapon used, parts of the body that were injured. Toxicology, that one was false. Alcohol. First step, a blank is identified with IR. The suspect blows into the device. Filter eliminates false positives. Measure the diffraction of light by alcohol is taken. Computer creates a reading. So those are the 5 steps. Kastle-Meyer is the first one that was pH sensitive. Iron and pH.
That's incorrect. It's just iron and pH.
So that would be luminol and phenolphthalein. gas chromatograph. That one is true. Is it blood, and is it human blood?
Remember, the skin turns pink.
Phenolphthalein, luminol fades quickly. Remember, that thing fades out quick. Phenolphthalein, and then Kassemeyer, microcrystalline, precipitin, and gel. So that's what we have to study up for the test. Make sure that you do some prepping, looking over stuff, and the test will be tomorrow. Hopefully it's not too bad. Take care. God bless.